Hello everyone, welcome to the Twisted DNA. So the topic for today is SDS Page, which stands for Sodium Dodecyl Sulfate Polyacrylamide Gel Electrophoresis. Yes, that's a mouthful. So now let's jump in by defining what is SDS Page. SDS Page is a technique that is commonly used in laboratory to separate proteins based on their molecular weight. And SDS Page utilizes polyacrylamide gels and an anionic detergent called sodium dodecyl sulfate or SDS, hence the name SDS Page. So let's take a moment to talk about this polyacrylamide gel that is being used in SDS Page. So the polyacrylamide gel is formed by the reaction of acrylamide and bisacrylamide, which results in this highly cross-linked gel matrix. We can see in this picture that when we combine bisacrylamide with acrylamide, the polymerization of these monomers forms this complex gel matrix, and the pore size of the polyacrylamide gel is determined by the concentration of acrylamide and the ratio of acrylamide to bisacrylamide. So the polyacrylamide gel used in SDS Page actually has two layers. So the top layer is called stacking layer and the bottom layer is called the resolving layer. Now let's understand the function of each of these layers. So the samples are loaded in the stacking layer. The stacking layer has a lower percentage of acrylamide and a pH of about 6.8. Since it has the lower percentage of acrylamide, the stacking layer allows the protein to move freely. And once the samples are loaded in the stacking layer, it helps the sample gets lined up so that it can enter the resolving layer all together at exactly the same time. And the resolving layer has more acrylamide and a higher pH of about 8.8. So once the sample enters the resolving layer, the gel then acts like a sieve through which the protein moves in response to the electric field, and resolving layer separates the protein based on their molecular weight. So I did mention the pH of these two layers, but we're going to look at the role of pH of stacking and resolving layer in a minute. So before we go into detail about how SDS Page works, it is very important to understand how the samples are prepared for SDS Page. So first, you take your sample and add something called sample buffer into your sample and mix the tube by flicking the tube. The sample buffer contains tris HCl, SDS, glycerol, beta-mercaptoethanol or BME, and bromophenol blue. Out of all these components of sample buffer, SDS and BME are the most important ones, and we're going to look at the role of these two in a second. Before that, let's talk about what is the function of glycerol and bromophenol blue. So glycerol is added in the sample buffer in order to provide density to the sample. So glycerol would help the sample stay at the bottom of the loading well so the sample cannot escape. And bromophenol blue is a dye that will help us in visualization of the samples in the well. So now that we've added the sample buffer to our sample, we have to heat these samples at 100 degrees Celsius for a few minutes, and you add the running buffer to the gel. So the SDS Page running buffer contains glycine, which plays a very vital role in SDS page, and we're going to talk about the role of glycine in SDS page in a minute. And once you add your running buffer, you load your sample alongside a marker, you turn on the power supply and run the gel until the dye reaches the bottom of the wall. And after electrophoresis is complete, you stain the gel using Comasi blue and visualize your results. Now let's talk about what is the role of SDS and beta-mercaptoethanol. If you remember, in agarose gel electrophoresis, the separation of DNA and RNA is possible because DNA and RNA has this phosphate backbone that provides both DNA and RNA a negative charge. But in case of protein, it's different. So proteins contains an overall positive or negative charge, and this enables protein to move towards the isoelectric point where the protein has no net charge. So therefore, this does not allow the proteins to be separated based on their size. Now, in order to separate a protein sample based on their size, we use SDS or sodium dodecyl sulfate before electrophoresis in our sample buffer. So why we use SDS is because SDS is an anionic detergent, meaning that when it's dissolved, its molecules have this net negative charge within a wide pH. Usually in the sample buffer of SDS page, a reducing agent called beta-mercaptoethanol or BME is added. So what this BME does is that BME is a reducing agent. So, this our protein gets this complex form due to this disulfide bond. So BME being a reducing agent, it is able to reduce the protein's disulfide bond, and so the protein can be unfolded into this linear chain. Whereas the SDS has this negative charge, which is able to destroy most of the complex structure of proteins. So the linearized proteins would now have a negative charge that is proportional to the polypeptide chain. So longer the chain, it's going to have more negative charge. Shorter the peptide chain, it's going to have less negative charge. Now, by denaturing the proteins and giving them a uniform negative charge, we are able to separate them based on their size when they migrate in the gel during electrophoresis. So in polyacrylamide gels, the smaller protein molecules will travel faster than a large protein molecules. Okay, so now that we've learned about sample preparation, I've also known the role of SDS and BME, let's talk about what actually happens when you load your sample and turn on the power for electrophoresis. Remember that the sample buffer has tris HCl, so the chlorine ion is negatively charged and it can move towards the positive end at much faster rate. And the SDS page running buffer contains glycine, which is an amino acid. So in acidic condition, glycine molecule becomes positively charged. At a neutral pH, the glycine ion is uncharged, so it's deuterion. And at higher pH, glycine becomes more negatively charged. So the pH of the running buffer is 8.3. So at the start of SDS page, glycine is negatively charged and forms glycinate anions. So when an electric field is applied, glycinate anions hit the stacking layer of the gel where the pH is about 6.8. So now the pH of two layers of polyacrylamides comes into play that I've mentioned earlier, that pH plays an important role in SDS Page. So what happens is, in stacking layer, glycine molecules are now mostly neutral. So they move slowly towards the anode through the stacking layer. Since chlorine ion reaches the anode at much faster rate than glycine, um, so glycine is still at the stacking layer when the chlorine ion hits the anode. And what happens is that this creates a narrow, steep voltage gradient between highly mobile chlorine ion and glycine. So the very high field strength makes negatively charged proteins move forward, and the proteins then form a tight band at the top of the dissolving layer. When the neutrally charged glycine cations hits the dissolving layer, they gain a negative charge, as the pH of the dissolving layer is 8.8. These negatively charged glycinate anions then take off towards the positively charged anode, and they speed past the protein layer. So now that the glycine gets past the protein are no longer in a very high electric field. So the proteins are now more relaxed and under the uniform electric field, where they move at their own pace and gets separated based on their molecular weight. So that is the principle of SDS page, and that's how the SDS page works. So that's it for this video. I hope you like it and hope it wasn't too long. Please don't forget to like, comment, share, and subscribe. And I'll see you all next time.